Welcome, everyone. It's nice to see you all this evening and beautiful afternoon on the coast of Maine. My name is Jerry Bowers, and I am the Director of Development and Public Affairs here at the MDI Biological Laboratory. And we are very excited to welcome you to our Science Cafe this evening. And I'm particularly excited about this evening's speaker. We have with us this evening Dr. Dorothy Klimis-Zacas from the University of Maine. She's a professor of clinical nutrition at the School of Food and Agriculture at the University of Maine, and also a cooperating graduate faculty member of the Graduate School of Biomedical Sciences and Engineering. So, as many of you know, when you tell folks that you are from Maine, people usually say two things: lobster and blueberries. And so tonight we're going to add a third thing to that equation, and that is science, because we're very excited. Dr. Klimis-Zacas is studying the role of the wild blueberry in potentially enhancing wound healing and tissue regeneration. And so many of you are familiar with some of the research that's currently underway here at the MDI Biological Laboratory focused on regeneration. And so we invited Dr. Klimis-Zacas here this evening to tell us a bit more about her work in understanding not only the nutritional benefits of blueberries, but also the potential role in enhancing wound healing and tissue regeneration. So Dr. Klimis-Zacas, we are so excited to have you with us this evening, and we really are looking forward to hearing your presentation. Thank you so much.
Great, thank you, Jerry. And please let me know if you can hear me.
Yes.
Great, thank you. And welcome, thank you for inviting me. This is a great opportunity to talk about the wild blueberries that are indigenous to Maine and to see so many people that that are here. So it's great to meet you and see you all. And today I'm going to talk about my most recent research on the novel role of wild blueberry extracts on wound healing and tissue regeneration. I have been working with wild blueberries for quite a while, and we have seen the amazing health benefits of eating wild blueberries, incorporating them in the diet. So this little berry, which is packed full of antioxidants, and is the Vaccinium angustifolium, the Maine indigenous berry. And as you know, probably, there are many different species of blueberries. We have the Vaccinium corimbosum, which is the high bush blueberry, the virgatum, the rabbit eye, grown in the southern part of the US, the Vaccinium angustifolium, the low bush here in Maine, indigenous and Eastern Canada, and the myrtillus, which is the European bilberry. So different species. I have been working with the Vaccinium angustifolium, the caviar of Maine, and and which is grown at the coast of Maine and parts of Nova Scotia, Prince Edward Island, and Newfoundland. So those are the berries I work with for my research. It's a composite from all of these different areas representative of the wild Maine blueberry. Wild blueberries are contributors to Maine economy, and the small wild blueberry farmers really depend on them. So this research also supports the industry, the wild blueberry Maine industry. So Maine wild blueberries compose about 9% of the pie, okay, of the North American blueberry pie. Canadian is 26%, and most of it, the 65%, are the cultivated berries, the high bush berries. Now, cultivated berries are really grown all over the world. And you can see here where the red marks are, okay, Europe, Africa, South America, Australia, New Zealand, and here in the US, okay? And their production has dramatically increased, and it's going up concurrently with more research. The more we discover about health benefits, the more the demand increases, and thus the production, okay? So here a little bit history about the Maine wild blueberry production and price, okay, that the wild blueberry farmers were getting. you can see historically, we see a slow growth and a sharp increase, okay, in the early 2000s, and production of wild blueberries. And then, as we can see, it dropped dramatically after 2017, and it's still low, and so are the prices the wild blueberry farmers are getting per pound. And that's for many reasons, because wild blueberries now are competing with all these different countries that are selling berries. And I can give you one example. Originally, I come from Greece, and I have research collaborations with Greece and Italy because I go to Milan and do my clinical research there. So when this research on the health benefits came out, the northern part of Greece really took out the citrus trees and planted blueberries. So you can see the same thing in Italy, okay, the corimbosum. You can see, and also another reason is because of the recent dryness, okay, of no rain during the last few summers here in Maine. So it's sad, but you know, things go up and down, okay, in circles or in cycles. So let's just hope that we will get this, you know, we'll get some experience from that and go on, and hopefully they will be fine in the future. So we call and categorize the berry, the wild blueberry or the blueberry, okay, as a functional food. So what is a functional food? Well, it's a food that has health benefits above and beyond basic nutrition, okay? So we're not just talking about the minerals and the vitamins and the fats, carbohydrates, and proteins, okay? The calorie providing it. These are the non-nutrient compounds, we call them, or bioactive compounds that the blueberry has, the polyphenols, that impart all these health benefits, okay? So all of the research that has been going on the last 20 years on the berries and in general and blueberries has shown that when the blueberry is consumed as part of a varied diet, but it has to be consumed on a regular basis and at specific and effective levels, okay? Like regular basis, and we see that over and over again, okay? And when we're talking about the berries, you can, as I have worked also with raspberries, and it seems that different berries are having different effects, beneficial effects on different organ systems. Maybe the blueberry is more potent or more effective on cardiovascular. And we just published an article about raspberries, the red raspberries. They are hepatoprotective, so they protect your liver, okay? So different berries, so if you have them, okay, every day, every other day, okay, consistently and a certain amount, they are very effective. I won't call them miracle foods, but what I see in my lab, okay, are actually, it's incredible, okay? So, blueberry nutrition, all right, blueberries, what do they have? Well, they're very high in water, okay, but they're very obviously low in fat and low in protein, and high in carbohydrates and very high in fiber. These are, it's about 100 grams, and 100 grams is about 1 cup, okay? And when we talk about vitamins and minerals, really they're poor in most minerals other than manganese, okay? And copper. And for vitamins, okay, vitamin K, the phytoquinone, okay, is about 16% of the daily value, and vitamin C. So they're not really very high, okay, extremely high in your so-called nutrients other than specific ones, but it's very low in calories, okay, very low in fat, no cholesterol, high in fiber. So if you want to enhance your fiber intake, you take the berries, raspberries, blueberries, okay? But what they have, and that what we have been studying, are the polyphenols or the phenolics. These are the non-nutrient bioactive compounds, which we have studied and on over and over and over again, and those with the ones that we have focused on and their health effects. Okay. So the wild blueberries, when you compare them, okay, with other berries and fruits, they have the highest recorded antioxidant capacities, which means that they help you from reactive oxygen species from the environment, depending where you live. Okay, so they protect you. It's like a shield. Okay, and it's the antioxidant capacity is correlated with the polyphenols and anthocyanins in the blueberries. Okay, we have the major anthocyanins. Some of you may have heard of them. Okay, which are common in the blueberry. And when we compare the total antioxidant capacity of the wild blueberry versus the cultivated blueberry, then you can see here it's almost twice. Okay, and the strawberry, the cherry. Okay, so it beats them all. Okay, so the wild blueberry, and we're fortunate to be in Maine and have all this wonderful access to the wild blueberries, although their price, as you might have seen, has gone up exponentially recently. Okay, and especially when you want to have the organic blueberries. Okay, because they are like sponges and absorb all of the chemicals, and their surface area is very high. Okay, so you want to have as much as you can the organic blueberry. Okay, now all those bioactive compounds, the anthocyanin flavonoids, account for about 60% of total polyphenols. So when we're studying the berry and the bioactive compounds, we're primarily studying anthocyanins. And anthocyanins, the blueberry has a dark blue color. It comes from anthocyanini, from the Greek word. Anthos is the bloom. Kyanos is the blue. Okay, so it's blue bloom. Okay, and that's what the pigment that imparts the color on the blueberry. Okay, they are catabolized, they're broken down very fast, and we find this in tissues, and their metabolites persist in the urine. Okay, hours, like 24 hours after we eat them. And another very important thing is you probably have heard all about microbiota, your gut bacteria, how it can change with diet. Well, depending on whether your GI tract, your gastrointestinal tract is healthy, depending on whether you have the healthy non-pathogenic bacteria that also change with your diet, well, that will affect how the catabolic products of the berries, how much of them are absorbed. So the health of your GI is very important. Individuals, as you know, with inflammatory bowel disease or other problems, okay, and depending on the bacterial species, they will have differences, okay, in absorbing and what they're absorbing. So the polyphenols, as we said, they're antioxidants. They're also anti-inflammatory. So we have much research showing that blueberries really decrease inflammation dramatically. They are anti-neurodegenerative and anti-cancer and many others. Any abnormal condition we studied, the blueberries corrected. It's amazing, okay? It is amazing. So these are the polyphenols we talk about, okay? And the blueberry is so complex. It has hundreds of different bioactive compounds, different structures. You can see we studied the phenolic acids, okay? We have tannins, and also it's also classified in flavonoids, but part of it is anthocyanins, okay? And flavonols and lignans and stilbenes, okay? So depending on their structure, they're classified differently, okay? So we are going to talk the different extracts that I have used from the berries would be the phenolic acid extracts and the anthocyanins, okay? So let me just tell you a little bit about, and I will go fast because we don't have very much time. my past research has shown beneficial effects on vascular function and structure and metabolism in animals that ate, had diets that had contained about 1 cup of berries, okay? And those animals were Sprague-Dawley rats, which are normal blood pressure animals. Then we use animals with high blood pressure. We use animals that were inflamed, the obese Zucker rats that liken the metabolic syndrome. And we found all beneficial effects, decreasing vasoconstriction. So like when the animals are stressed with a phenylephrine, stress hormone, their arteries are like loose, they don't contract. Very good. So that means their vascular function is normal. They don't become dysfunctional. Also, we had clinical studies with humans that had multiple risk factors for cardiovascular disease. When we fed them the berries, their vascular function improved, okay, which is wonderful. And then I worked with these animals, the Zucker rats, that are a metabolic syndrome. Metabolic syndrome, I don't know if you know what it is. These are individuals, when we gain weight, that new adipose tissue, it's like another organ. It becomes inflamed and sends signals to all the different organs in your body to say, to inflame them, all right? So these animals have inflammation, have high blood lipids, they have insulin resistance, they have everything. And when we work with them, we find that feeding them 1 cup of blueberries for 6 to 8 weeks, it just modifies and normalizes everything. Their inflammation decreases, their blood lipid profile becomes normal, glucose metabolism becomes normal, and the gut microbiota become from pathogenic, the non-pathogenic increase. So it's amazing. Okay, we have published all that. Don't look under my name if you're interested, and then you'll find the presentation. But for today, I just want to go over what happens when these are not diets that I'm going to talk about. These are just extracts from blueberries, and we're not using animals or humans to feed them. We use cells, okay? And these cells are human endothelial cells that line your blood vessel, okay? Now, and we're studying how cells migrate and how or do they form new blood vessels. So we're studying angiogenesis, which is the formation of new capillary blood vessels from pre-existing ones, okay? That's what angiogenesis is, the normal biological process, and we need angiogenesis for wound healing, for wounds to heal. Okay, also for atherosclerosis, for ischemic condition. If somebody has a stroke, they develop, we want to help that tissue perfused with oxygen. So we want new blood vessels to form, okay? So ischemic conditions, and for cancer, of course, we want to stop that, arrest it, okay? So in some cases, we want to promote angiogenesis, and others, we want to arrest it. Okay, now, so this is what happens when there is some high blood pressure, shear stress, problems with low oxygen. That is a stimulus for cells to start migrating, okay? The cells migrate, they divide, they differentiate, and form a new blood vessel if it's needed, okay? For the cells to perfuse that tissue, for oxygen to come, for new blood vessels to form, okay? Like in wound healing. And so cell migration and angiogenesis are related, okay? And they're associated with growth, with cardiovascular disease, with atherosclerosis, with low oxygen, and wound healing. Okay, I'm not doing that. And we have some proteins that we studied that help cells migrate, raw and RAC. And then we have some more proteins that, and this is all interrelated to promote cell migration and new blood vessel formation, but I'm not going to talk. So in our experiment, so now I'm going to get into actually what happened, okay? We wanted to investigate what will happen if we have some cells and we expose them, the endothelial cells, the ones that line the vessel, and we expose them to different fractions from the blueberry, anthocyanins and phenolic acid, and also a combination of the two. Are the cells going to promote migration? Is migration going to be promoted? Are they going to form new blood vessels? And so we studied the speed of endothelial cell migration, and we studied angiogenesis, whether they will form new blood vessels, and also studied the gene expression of proteins that are really, really associated with migration or angiogenesis. And I will go through very fast with that. We use this HUVEX, the endothelial cells, and we plated them and grew them. We formed the growth curve, studied that, and then we added different concentrations of our extracts to see whether they were going to proliferate or whether they was cytotoxic. So we knew the concentrations. The wild blueberry powder, the Wabana, Wild Blueberry Association of North America, always sends the picks, and pulls the blueberries from these different areas marked in red, and sends them to this company Futureceuticals that is freeze dried, and then it's delivered to the researchers. Okay, and then from that blueberry powder we extract, and we have two fractions. In our case, the anthocyanin fraction and the phenolic acid fraction. And we determine concentrations of phenolics and concentrations of anthocyanins. And with HPLC, we fingerprint it, as I mentioned before. Then how do we study cell migration? Well, we have this EB dish that has two kind of containers and a cell-free container in the middle. So we seed the cells in these two containers, and then we expose them to different concentrations of our extract and measure the speed of cell migration. And then how do we measure whether new blood vessels are formed? Well, we have this other microslide, and it has a matrix that promotes growth of the blood vessels. And then we add the cell medium, we add the cells, and then we add the different extracts and see what happens, whether they will form new blood vessels. And we study, so you can see here, we study the different, we call this meshes, this would be like your tubular network, okay? We study the number, the area, the master junction, the nodes, okay? So that's how we assess angiogenesis. And the gene expression of proteins that are very, very critical for migration, rock and roll, and proteins that are very critical for angiogenesis. Okay, and I'm gonna show you the result. So this is the HPLC profile. This is our fraction of anthocyanins. Again, you can see there are many different types of anthocyanins. And our phenolic acid fraction, it has only chlorogenic acid, which is good because that's what we're working with. And here are the composition of the anthocyanin fraction, which we have. A lot of it is malvidine glucoside and many others. I'm not going to bore you with that. We can go back if you like. And then we are going to add different concentrations on for our cell migration assay. So these concentrations, so we're going to add from 0.002 to 120 micrograms per ml. So these concentrations, the first three, are biologically found in the bloodstream normally, okay, when we eat the berries, okay? And then we add the combination of anthocyanins and phenolics. And so this one is the first. I want you to pay attention to this. This is the first little clip I have here, and you see to the left, I'm going to start it. You see how the cells migrate. To the left is the control. To the right is the anthocyanin fraction. And you can see how fast the control is moving to close the gap, which mimics the injury. What's going on? I don't know what happened. Okay, here, I won't touch it. And you can see to the right of your screen, how much slowly the anthocyanins, okay, the cells that have been already exposed to anthocyanins. The control already closed long time back. So you can see the anthocyanins arrest the migration of the cells. And when we quantify the migration rate, we can see that the particular concentration is significantly lower than the control, okay? Like the picture showed us. This one is when we expose the cells to the phenolic acid, okay? So look at that, the control to the left, the phenolic acid to the right, and look how fast they go, boom. Okay, so that was really surprising. Nobody had really shown that before, okay? And when you quantify that, then you see, look at it, in so many different concentrations, control versus the phenolic acid, significantly higher migration rate, okay? So that made us happy. And we said, well, let's see now what will happen, whether this cell migration led to higher angiogenesis, okay? And when also we did the combination treatment, anthocyanins in equal amounts to phenolic acid, that also increased the migration rate. And here is the kind of collage that I put together. You see the control, 8 hours, okay, it's all closed. Anthocyanins are still going. Phenolics also probably by 7 hours are closed, and the combination by 8 hours is closed, okay? So you can see, so we thought, gee, this is interesting. And we continued to study the angiogenesis. So now we chose only the concentrations that we found significant with anthocyanins, phenolics, and the combination. And again, we measured these meshes, okay, which are the tubes, the tubular system, which represents the blood vessel formation, the number of tubes, the area of the tubes, these segments, and the nodes, okay? And so look at this, when we are adding and exposing the cells to the anthocyanins, look at that. This is the control to the left. I think we have, hold on, let me see. I cannot see them because, so anyway, the left is a control. It's a beautiful tubing system. And look at this, it's like it fell apart, okay? So in any condition that you don't want any new blood vessel formation, like in cancer, you don't want to feed the tumor, well, there, I can use the anthocyanin extract, okay? And so you can see here, number of meshes decreased, total mesh area decrease, so we can see it cannot form an integral, okay, the anthocyanins. Now, look at when we expose them to the PA, the phenolic acid fraction, you see, look at that control, and look at that, they're able to form a beautiful network, okay? And we were very happy with that because that went along with the migration of the cell. And you can see number of meshes increased, total mesh area increased, number of nodes, number of master segments, and number of master junctions, okay? So we're happy with this data. And when also we add the combination of the two fractions, again, they are able to form, okay, all the beautiful network, tubular network. And when we quantify, we see that we have a significantly increased total mesh area and total master segment, okay? So we were happy with those results, and we summarize again, control, ACNs fall apart, and phenolics and the combination promotes angiogenesis, okay? So I'm not showing you any gene expression or any protein expression, but the conclusions were that the anthocyanin fraction has an inhibitory effect, while the phenolic fraction from the wild blueberries significantly promotes endothelial cell migration and angiogenesis, and combination also does. And along with that goes our gene expression of the two proteins that are important in migration and angiogenesis. They're all upregulated, mean they increase with the phenolic acid fractions and combinations. So it all agrees and makes a beautiful story. And what is the significance? Well, it's the first time that we observe that there is differential response from blueberry extract for cell migration and angiogenesis, and it can have many different applications to different medical conditions, wound healing, diabetic foot ulcers, burns, ulcus decubitus, or any condition benefited by promotion of wound healing for the phenolic acid wrap. And we have published this, okay, 3 different articles. You can go on, find my name or Panos' name, and you can read them, okay? And we studied the pathways by, we know how the berries are doing that, okay, through which pathways. And so we went ahead. So this is what I want, if anybody is, you know, faculty member or student or whatever who's, you know, watching this. So, you know, as scientists, we usually never think about, you know, going from bench to, we do try to go from bench to bedside, of course, but you have to go through the market to the bedside. So we decided to, let's protect ourselves, and the University of Maine applied, we applied through the University of Maine to be protected by this patent, which is international, with me and my graduate student, my doctoral graduate student, Panos Sakiroglu, who is now postdocing at the University of Maryland School of Medicine. And we are all collaborating still on this, because the next step was for us to become entrepreneurs and to go from the bench to market, to bedside through the market. And so we said, okay, what's the next step? The next step was for us to start conducting and proving that, actually confirming these results, which were done with cell culture with animals. So I will go on to talk to you a little bit about our animal studies, the preclinical studies. So the berries, as I mentioned, the freeze-dried berries, the extracts of anthocyanins and phenolics, and we have our model is the Sprague Dawley wrap, and we create 4 dorsal wounds, and we apply on those wounds daily, okay, the extracts in different concentrations. So we apply the phenolic extract and the mixture of phenolics and anthocyanins, okay? And this is the setup, the control, which we put nothing on the wound. The phenolics at 250, 500, and 1000, and the same thing with the mix, okay? And we have our surgical suite. So this was just about impossible to do during COVID. I had to rewrite all kinds of protocols for distancing, the masks, and you can see here Dr. Weber, who is our veterinarian, and wearing. So we bought this. This is a welder's helmet. Actually, I got it from my dentist who had it. And then I thought, oh, good. This is, you know, we have to do this experiment. So we bought it for Dr. Weber. And it was fine. And we had like 2 people in the surgery room. Then post-surgery, we had like 1 person, another person. So we were really distanced. So we were able to conduct the study and get some results. So these are our Sprague Dawley rats under anesthesia. And my student is actually preparing those wounds, creating the 4 dorsal wounds. And we apply right on the wound. We apply the extract, like a different concentration. Each rat gets like the control, the extract, concentration 1, 2, and 3. And so we have 4 samples here. So this is control day 0 and day 6. So for 6 days, once a day. And these are the phenolic acids. And we're finding the best response with the phenolic acid at 500. And this is day 6. So when we calculate, and we just want to see, and we photograph these wounds and measure them, and daily, okay, with a digital camera. And then we do macroscopic analysis of these surgical wounds. And the photographic data, then we do the wound with the, assess the percent closure using image J. And so this is we were very happy to see that, because with percent wound closure, we're measuring, this is the control, and these are our extracts. So we're doing a 20% above and beyond, which is wonderful. With the cells that we cultures, it was 38%, because you have the cells and you expose them right there. But here you have the skin. So all the compounds have to go and be bioavailable. And so we were very happy because most products in the market, they're only 6% wound closure, okay? So really low. So we have an advantage. This is 20% above and beyond. And then this is the control the mix, okay? Also was statistically significantly higher, okay? Then the next step that we're doing right now, we're measuring genes of interest for inflammation, because this is anti-inflammatory, bronchiogenic, and also anti-fibrosis and vascularization. So we are doing the gene expression, we're doing ultrastructure after staining them and semi-quantification. So we're studying vascularization of the tissue. And we're also doing protein expression in markers of interest that show and depict fibrosis and vascularization and angiogenesis. Also, the hypothesis is that these extracts have antimicrobial activity. So we're also checking for antimicrobial activity and with different assays for against different bacteria that reside commonly in wounds, okay? So our summary from at least preliminary data from the extracts are that the wild blueberry phenolic extract at this particular concentration in crude increase wound closure by 20% above the control and have the potential to reduce inflammation and have an antimicrobial role. So we are happy with this and we're continuing now. And we have been involved in tandem. We have been involved with the Office of Innovation and Economic Development at the University of Maine. And all these individuals have helped us a lot learn business, you know, and for the last 2 years. And we have been involved in entrepreneurship, whereby we have been involved with the National Science Foundation, the ICOR program, where we did extensive customer and market discovery. And also we have been involved with the Maine Innovation Research and Technology Accelerator, whereby we explored areas of commercialization, different industrial partnerships, prototype development. Actually, we have developed our prototype, cream and gel, and we're testing it right now, manufacturing and scale up. So hopefully we'll be able to go to market pharma, biotech soon and go to market by early next year. And Maine Innovate Series workshop, and we took some workshops through the University of Vermont, funded by the National Institutes of Health. So yeah, we've done a lot of work in the last few years. This was the team, NSF ICOR team 2019. That's when we started, and there were many, many hopefuls. We had like a cohort of 5. So the University of Maine continues that. I think we were the second cohort. This is my team. They're wonderful, my wonderful students. And Panos, that his PhD thesis was on this. And Natalie, who worked with the raspberries, but she's a great business person. So she's going to be helping me. She's getting her PhD in August. She's just doing her dietetic internship, and she will be involved in the company. And funding came from the USDA, the Wild Blueberry Association of North America, that I thank them. They've been funding me the last 20 years. The Maine Institute of Technology, Maine Technology Institute, the University of Maine Medicine Seed Grant Program, which was really important to get us started, get us off. The Maine Innovation, the MERTA, and we are going to be joining the National Science Foundation, the National ICOR program for more customer discovery, and et cetera, et cetera. So there are many students that I'd like to help. And my dear colleague, James Weber, who's part of my team, and Christy Townsend, who's not anymore at the University of Maine, sadly enough, and Magda, Panos and Natalie, and a lot of the students are nutrition, FSN students, or the Graduate School of Biomedical Sciences and Engineering. My new student, Esther, who is analyzing the data on these studies right now. She's a doctoral student. And the undergrads from the Food Science and Human Nutrition, the undergrads from the AVS, the Veterinary Science Program, and the Small Animal Facilities personnel. So I thank them, and where I can see my lab is all women. So I don't know if that means anything, but so I will keep quiet now and then get your, thank you so much. I hope you understood something or learned something today. And now we're open to questions.
Absolutely.
Yes,
This was
Jerry.
fantastic. Thank you so much. That's a really,
Sure,
really
you're
exciting
Thank you.
story.
Yeah, well,
Exciting for you, but also exciting for Maine. I think it's a really fun thing. So, okay, we're going to open up for questions. We have a couple of questions in the chat. I just want to get to those first. And then for those of you who want to ask in person, if you want to use the raise hand feature, which is at the icon at the bottom of your screen. I see some of you have your hands up already. Dorothy, we have a question from John. He says, number one, he's asking about organic versus non-organic and saying that, you know, sometimes it's hard to find the organic. Do you find that to be true, that it's a little more, they're a little more difficult to come by in the state of Maine?
Well, yeah, you kind of have to know where to look, you know, and yes, definitely in the supermarket, you won't find, you know, the organic. At least I have not, you know, Shop and Save or whatever. I hate naming names, you know. But yes, we look for the organic, you know, like, that's my philosophy. You just don't want, in general, and even with berries, with the larger surface area and being like sponges, and with any food that we put in our mouth, we try for it as much as we can to be organic. We don't want to bioaccumulate all this pesticides, insecticides, herbicides, et cetera, et cetera, et cetera. Now, where do we find them? Well, we find them in the health food stores in Bangor, in Belfast. So wherever we are, you know, on our way, we have a deep freeze and we pick them up. And, you know, I have observed their prices have gone above and beyond. They used to be $25 for a 5-pound box, and now it's $35. So I don't know when they will stop being affordable. You know, so it's always like risk versus benefits. What should I do? Should I bioaccumulate all this and yet have this beneficial effect, you know, of everything we talked about? You know, it's hard. But yes, we should just try to have the organic, definitely.
So you mentioned freezing them. Do you see any difference in fresh berries versus frozen in terms of the health?
Yeah, so okay, so fresh is the best. Okay, there are ways that you can even increase, you know, bioavailability of what's in the fresh. It's called this tunnel kind of poaching that we have in the lab, but that's lab stuff. So I would eat, number one, I will do the fresh, and number two, I will do the frozen, you know, because that's, you know, it's always good to have something instead of having nothing. Okay. Also, you know, we found that with, at least with the powder, that, you know, you really should not keep it, because we usually, our powder is vacuum packed, you know, so we should not keep it. And we have an article on that, longer than, like, I wouldn't use it longer than a year, like for my experiments. I want to have fresh as possible. But for eating, you know, like frozen berries, well, as close to the production as possible, you know, but how do you know? You know, the other thing is, how do you know when these were, you know? Usually look in the back, and I haven't even looked to see whether they talk about which crop it is, you know, like which crop. Usually when I do my research, I do not allow them to give me from crop of last year, let's say. So like if I'm doing research this year, I want to have berries from the 2021 crop that they pick in August, and I can have available in December. So as fresh as possible. Another thing we found, based on the year, you know, those bioactives, those polyphenols, they vary. It's like amazing how much variation you can get, because like, so, you know, your results can differ, you know, but anyway, we just look at the global picture. So As long as we can have the fresh, yes, but the fast frozen are also fine.
Fascinating. That's great.
But the less processing you do, the better. You know.
So what about dehydrating berries? I mean, I know I've seen more and more of those lately, it seems like. Does that have?
Well, I mean, it just depends on, the heat, the temperature. And so I would not want to have too much, very high heat, depending on the dehydration process, what the standards are, what's the method. And I have not seen, there must be, but I haven't seen, which is a good question, any articles comparing, the bioactives within the dehydrated berries. Maybe there must be something, but I have not seen it. Yeah.
Okay.
But I would think they would be lower. Okay. But also like breaking the skin, of the berry, it makes the or poaching them, like throw them in boiling water, taking them out, that increases bioavailability also. So there are many different ways. Yeah.
Okay, interesting. So one more question in the chat, and Stephanie asks, we're familiar with sort of the anti-inflammatory, antioxidant benefits of blueberries. Do you see any indication that blueberries have an impact on lifespan? Have you seen any?
The aging.
Yeah.
Well, they would. I mean, there are some of my colleagues are conducting studies on aging per se, but indirectly. Let's say, for example, an individual has metabolic syndrome, is obese, blah, blah, blah. All those are comorbidities. They decrease, lifespan. That's so, the blueberries indirectly are going to. But on aging, so, telomere length, size, or in general, I have not seen many studies on that, but I know that they are working in that area. Definitely.
Fascinating. So Alan, do you have a question? You can unmute yourself and ask if you'd like.
Yeah, it's just the Alan. Let's see. All right, I can't see myself, so I hope I'm not.
Right, I can see you.
I love the accelerated wound healing due to angiogenesis and cell migration, but those are also the same mechanisms for tumor growth. So yes. Is this something that if you have cancer, maybe you shouldn't eat a lot of blueberries?
Yeah, exactly. Yeah, that's exactly what I said. So it depends. So that one extract with the anthocyanins, that's pretty good because that is going to stop those blood vessels on their tracks and those, cells. Okay, that's good. So you can have the extract. Okay, but for wound healing, and that's the whole point, the extract for phenolics that will be pro-angiogenic. So we're not talking about eating the whole berry, but if you eat the whole berry, putting all those together, there are some studies, about the whole berry that they are angiogenic. Okay, so it's good for people that have cancer. Okay, so what I talked about were when you start extracting and separate those extracts. So one extract would be good, and actually it can be used clinically for cancer. Okay, and the other extract will be good for wound healing or atherosclerosis. Okay, cardiovascular disease, somebody who has a stroke. Okay, I would like to give it IV. Okay, to build new blood vessels and accessory circulation, but that would be like 20 years down the line.
So my has a question. She wanted to know if you have a wound, should you mash up blueberries and spread it over the wound? Or
yeah, well, because did you see how the blueberries have so many different components? Okay, so and they work either synergistically or antagonistically. So I wouldn't know what the end result would be and how much of that would be absorbed. I can only talk about my fraction that has the chlorogenic acid in it. So needs to be extracted. And that's why we are creating this product for that purpose, for just for wound healing. Yeah.
Wow.
Good question. Very good. Yeah.
Thank you.
Alan.
Sure.
So, Dorothy, I have a question for you about the, your company. So you mentioned that you have a patent and you're going to produce what I think you said was a topical treatment for wound healing. Is that right?
Yes, yes.
And so, but for cardiovascular disease, do you envision an oral application for this as well? How are you?
For cardiovascular, what I'm thinking, I don't know. I mean, there's so many spin-off products that I have in mind beyond, like many things. I cannot reveal them here.
Yeah, sure.
But for cardiovascular disease, I was thinking more of stroke, okay, patients that really need that accessory circulation and extra perfusion. And I was thinking, no, you wouldn't do it by mouth. It would have to be IV targeted. Or, maybe encapsulated with something, because when you do it by mouth, it has to bypass the GI, and there's so many obstacles there, and then it gets diluted in the bloodstream and then gets to the target. So the easiest way would be IV, okay? And there's so many factors, when you take anything by mouth.
Okay. All right.
Yeah, hundreds.
And another question that just came in the chat, and we were talking about this a little bit before we started, but Anna is asking, some of us don't have access to wild blueberries. What is the difference when you compare cultivated blueberries to the wild blueberries? And I saw one
you mean to get cultivated, organic? Is that what she's asking?
Yeah. Is there health benefit? Do you see a dramatic difference?
Yeah. Well, I have, where is it? If you let me share, can I share?
Yeah,
you're screen sharing. It's okay. So I'm gonna go down and take a look. I have the comparison. I had it at the very end. Of the compared here. Compare the wild blueberries with the tradition.
Cultivated.
High bush. Okay. So the wild blueberries have 72% more fiber, 2 times the antioxidants, like I showed you, and the anthocyanins. So yes, they're more compact, and they're higher in concentration, so it's good to have the wild, but it's also good to have, the low bush. It's fine. But it's good to be able to have preferably the organic ones. Okay. So if you can find that, that's the thing. I always worry, about, even here in Maine, where the farms are, the organic farms, and what is around that farm. Is there a mill? Is there, I always put the map down to see where the farm is and what else is around there, just because they are so like sponges. Don't pick up anything.
Yeah.
So, it's like risk versus benefit. It's always, and we have to balance things out. I'm just telling you the ideal situation here. So what it would be. So maybe have everything else organic and have the blueberries at there.
Right.
Not,
Right.
So
do you eat a lot of blueberries yourself?
Yeah, every day. I have about a cup in my frothy, I call it, with my soy and it's good to put an acid environment, so like some orange juice to promote bioavailability. Yeah, every day. It's my kind of functional drink.
That's great. So you talked a little bit about forming your company. What about your research in general? What's next for you in this project?
Yes. So what the next, my graduate student right now. So we are developing, we want to improve bioavailability as much as possible and make sure, it's because there are 2 concerns. Okay. So right now we have this one product that has pretty good, I didn't show you the results, but it's pretty good, the one prototype, which we are going to, we have pretty good results with that, but I want to improve it, improve the results and have a totally kind of new product in the market. And so the next step is to create something which is encapsulated, which is like a nano delivery, which will be make sure that the bioavailability, it gets slow released into the tissue, and it protects all those very, very sensitive compounds. So that's what we're working on, developing something encapsulation that will, so that's what my graduate student is working on and testing it. So next fall we'll be testing yet another product and comparing it to what is in the market, because right now in the market, I tell you, there is nothing. There is just the triple antibiotic and the, the petroleum jelly. There's hardly anything. So our product will be helping, promoting wound healing, being anti-inflammatory, antibacterial, and regenerative. So
I've heard people say that Manuka honey has healing properties like that. Have you heard that?
Yeah, there is. Yeah. Well, I mean, you read all those things. So antibacterial supposedly.
Okay.
But it's not, it doesn't, maybe anti-inflammatory. I don't think it, I'm not sure about promoting the cell migration and angiogenesis with the Manuka, because we searched everything in the market, but it does not have all this kind of package
in one thing,
of, benefits.
You also mentioned the great variability just in the wild population of the blueberry, and like how you have to be careful about that. Is there any research underway to maybe do some either genetic engineering or breeding of specific blueberries to emphasize those qualities that you're looking for?
Oh, yeah. Well, yeah. Well, yeah. Well, being wild blueberries and indigenous to Maine, I don't know how much you want to change them, and that's the whole selling point. So yeah, it's a good idea. Maybe the high bush blueberry people want to do
Yeah, there you go. Well, listen, this has just been fascinating, and I've learned a lot about things I never knew about blueberries. So thank you so much, and we'll look forward to having you back when you get your company and your product on the market.
Oh yes.
Love to hear more
Thank you so much. It was great to see you, Jerry, and great to see all of you. I hope you learned something.
It was wonderful.
Thank you very,
very much.
And if you need anything, let me know.
All right, we'll do.
Okay.
We'll do.
Okay.
Wonderful.
Bye, Emily and Carrie. Bye.
And for those of you who are still on, we want to just remind you that our next Science Cafe will be on Monday, June 14th, and that will be given by James Wright, who wrote a biography about the real James Bond. And those of you who are bird watchers, you may recognize that name. James Bond was an ornithologist who spent summers here on Mount Desert Island. And so we're excited to have Jim with us to talk about the biography that he wrote. So we hope you'll all join us, and thank you again for your continued support for these science cafes. Without you, we would not be able to make them possible. So we really, really appreciate all the loyal Science Cafe attendees and supporters that we have. So thank you all so much, and we look forward to seeing you next time.